Thank you, Therese. So my name is Danielle Scott, and I am the Chief Officer of Product Development at GlycoPath. GlycoPath is an early-stage biotech company that is focused on commercializing and developing assays for the prevention and prevention of recurrence of cancer and other infectious diseases. So the main problem that we are focusing on right now is liver cancer. Liver cancer is a progressive disease. Progressive meaning that once you reach that fatty liver point of disease, there's very, very, very slim chance of turning back. And as Americans, we all love to drink and eat. It was mentioned earlier, prostate cancer. Fun fact, in South Carolina in 2020, liver cancer surpassed the number of cases of prostate cancer. We have all kinds of fun statistics up here on the slide, but eventually, as liver disease progresses and if it becomes hepatocellular carcinoma, the annual treatment cost can be around $20,000 to $35,000 per year, thus enhancing the need for socioeconomic and lifestyle need for a better, less invasive method for treatment. So our solution is our Glycotyper assay. Our Glycotyper assay is a proprietary liquid biopsy assay that is centered around utilizing N-glycan profiling to provide substantial improvements in diagnostic assays and prognostic therapeutic prediction assays. We use a liquid biopsy technique, so we draw serum and use that. This is incredibly non-invasive and results in a biosignature of captured proteins from the serum that can be considered intellectual property. This covers a variety of marketing segments, and we've recently shown from our GlycoFiber-Typer assay that we have surpassed current standards of care. So our assay is able to differentiate between fibrosis and cirrhosis with a 95% accuracy, from cirrhosis from healthy patients with a greater than 95% accuracy, and differentiate between low-grade and high-grade fibrosis with a greater than an 80% accuracy. Currently, the standard of care for diagnosis of liver disease is a liver biopsy. A liver biopsy involves a very, very, very large needle going into a person's liver and pulling out a very, very, very small portion of tissue. And that is supposed to give you a diagnosis that is a representation of the entire organ. By using our technique, by using single serum, we can obtain even more information on the global state of the liver from a much less invasive technique, such as a blood draw. Our current funding has come from STTR grants. So in our first grant was for the GlycoFiber-Typer, which I had previously mentioned. That was funded in 2019, and we recently published the data on that. That was the data that I highlighted earlier with the 95% accuracy. Starting next week, we will be getting a phase 1 grant for our expansion of our Glycotyper assay, which is the GlycoHCC-Typer. This is going to be an expansion, expanding our assay into the hepatocellular carcinoma state. Once we go for our phase 2 of that grant, we plan to expand to NAFLD and NASH. Additionally, last year, we received a seed funding from Brooker Corporation, which is the company that manufactures the mass specs that we use to analyze our assay. And this was a unique and incredibly fantastic opportunity for a partnership. And so we are very, very glad to have them on our team. So our current business model is around the SBIRs, STTRs, and our funding from Brooker. We also have launched our basic N-glycan kits. So these are research-based kits that have allowed us to enter the glycan research community, sell to researchers, and encourage them of the utilities of glycan research and how that can benefit their research. Additionally, in the future, we hope to become a CLIA lab and sell directly to physicians. So just a brief overview of the market. There are currently 5.4 billion patients with either HBV, HCV, NASH, or cirrhosis. Counting that as our total market, if we charge approximately $300 per test, which is comparable to the current cost of an AFP test, at a 10% market penetration, that would equal over $162 million in annual revenue. So who are our competitors? What else is out there? So first, I'll mention the AFP test. So the AFP test is an alpha-fetoprotein blood draw test that measures the levels of alpha-fetoprotein in your blood. The biggest drawbacks of this test is it has low sensitivity, low specificity, and when combined with ultrasound, 1/3 of the time it fails to detect early HCC. Oh, and I forgot to mention, it's not for cirrhosis or fibrosis. Next, we have FibroScan. So FibroScan is a assay that performs a transient elastography that generates a one-dimensional, i.e. a line, version of the stiffness of your liver. That has about an overall 82% accuracy. Finally, we have FibroSure. FibroSure is also sold under the name of FibroTest, and it is sold under that name through LabCorp. It is a biomarker blood-based test that factors in biochemical markers, age, and gender to generate a score that is representative of the overall state of the liver health. This also has a much lower accuracy than our current test. So what else are we doing? Because we have this mass spectrometry-based imaging platform, not only can we apply this to liver fibrosis and cirrhosis and hepatocellular carcinoma, but we can also apply it to a variety of other disease states. Through our academic labs at MUSC, we have done some work that we are hoping to translate into some additional phase 1 grants that include looking at changes in transplant patients, lupus changes, and Lyme's disease diagnostics. So who's the team behind this? Who's leading us? So first of all, we have our acting CEO and one of our founders, Dr. Richard Drake. He has been in the imaging mass spectrometry field for over 30 years and is a key thought leader in the field of glycomics. Next, we have our CFO and another founder, Dr. Anand Mehta. He is also a professor at the Medical University of South Carolina, and he has several companies, including one that manufactures the PNGase F enzyme. This is a key component for us because this particular enzyme is what releases our glycans that allows us to do what we do. Without this, this enzyme is incredibly expensive. We wouldn't be able to do what we do. Next, we have Dr. Peggy Angel, who is our acting chief scientific officer, another founder. She has plenty of years of experience in academia and industry and knows the ins and outs of getting a mass spectrometry assay to market. Then we have myself. I function in day-to-day operation, instrument management, and have had a lot of experience in the lab and getting things commercialized and marketed within the scientific field. Last, but absolutely, definitely not least, we have Dr. Steve Castellino. He was a former director at GSK and has worked in their MALDI imaging group and is just an all-around fantastic addition to our team. So how do we know this is going to work? How do we know we're not going to flop? How are we going to not stay in that valley of death that Dr. Doloff referenced earlier this morning? So first of all, the majority of FDA-approved biomarkers are glycoproteins or glycans. And changes in glycosylation have been measured since the early 1960s and have been noted as changes that occur within various cancers. Additionally, our assay works on a variety of mass spectrometry platforms, and the glycan panels derived from our analyses are portable and can be used and have been used in thousands of samples analyzed by GlycoPath founders. And finally, this uses mature antibody slide array technology. And finally, what is our exit strategy? How are we actually going to make money out of this? So eventually, our goal is within the next year, hopefully, to do a Series A round of funding that will aid us in getting to that CLIA lab and begin our commercialization techniques. But when it comes to an actual exit strategy, we hope to be able to do some form of an M&A in between June 2020 and June 2021. There were 288 mergers and acquisitions within the medical tech sector, generating over $85 billion. And so we hope to also be a part of that one day, but we're not quite there yet. So with that, I want to thank you for your time, and if anyone has any questions, I would be happy to answer them.
I've always had the impression that liver cancer is very aggressive and fast-moving.
Yes.
So how symptomatic would a patient have to be to have your test, need your test, or have an indication that the test should be given? And is it really too late for you to have an impact? Yes, it's a simpler, better method, but is the patient too far along for you to have an impact on longevity?
No. So especially the Glycotyper test specifically is aimed at diagnosing that stage 1 fibrosis to cirrhosis phase. Those happen before you even get to the liver cancer. So within that, just that Glycotyper platform, we're hoping to be able to continuously monitor and diagnose as opposed to every 6 months. So right now, standard of care, if you begin to look like you might potentially have or be a candidate to develop cirrhosis or fibrosis or cirrhosis, they want to do monitoring every 6 months. Well, that means every 6 months you have to have a very large biopsy needle go into your liver. A lot of patients aren't going to be compliant to that. A lot of patients are much more likely to be compliant to a blood draw. And so if we can continue to monitor that and actually get the compliance of every 6 months, the patient's going to be a lot better off as opposed to saying, okay, you have to go in every 6 months for a liver biopsy. Well, I'll wait. I can wait a year, maybe 2 years. If we can catch it earlier within that patient compliance window, we can then treat the patient better and ward off the potential of hepatocellular carcinoma before it happens.
One quick question. No MRI or CT scans studies work to diagnose?
So you can, but those are incredibly expensive. And again, your patient compliance goes down. A lot of those studies have also had mixed bag of results. And so a lot of doctors have shied away from recommending those modalities because of the mixed bag of results that they get.
Can you explain a little bit more about your regulatory strategy, and in particular how CLIA is going to lead to getting the data? Because I would think you would want to be a certified test versus just CLIA is your end goal, correct?
So right now, we're just focusing on the CLIA certification because we are a mass spec-based assay. So it's not quite as easy to just directly integrate into something such as Quest or LabCorp. But eventually down the line, as the mass spectrometry platforms continue to grow and as more assays are available within those platforms, we think that it will eventually grow to that. But right now, we're focusing more on the CLIA lab designation.
How did you determine the price point?
So right now, that is based off of just kind of an average of what's on the market currently and then what we are spending to run all of our samples. It is not by any means a definitive number, and I cannot get you the exact profit margins off of it because we're still in the research and development phase. We're still optimizing. But that is the best rough estimate that I can supply you with today.
Great.
With the CLIA strategy, like how do you, like would samples get sent to you and then you would process them in the CLIA lab? Like how do you deal with a more national footprint with that strategy?
So the goal would be to have regional labs and then have samples shipped to them within particular regions.